Welcome to Chapter 18 and another series of narrated slides. Today we're going to talk about digestion, the physiology, well, kind of the physiology of digestion, and the regulation of feeding, why you select certain foods, hypothetically. All right, first part is you and your digestive system. It is very interesting that digestive systems of most animals are at least anatomically similar. There's a mouth, there's an anus, and food gets digested, broken down somewhere in between. Humans are no exception. We use our digestive system to, one, break down larger molecules into smaller molecules. For example, proteins into amino acids. The second part of digestion then is to absorb these smaller pieces back into the blood so it can be utilized for various reasons by your cells. This is just a series of chemical reactions showing the breakdown of three of our major nutrients, carbohydrates. So we're taking something like starch, which are multiple glucose pieces glued together, proteins made up of amino acids, and of course very long chain lipids that we can break apart to form individual fatty acids. The human digestive tract, again, begins with a mouth, ends with an anus, and is one of the ports of entry of microorganisms, including you know what. So the digestive tract must allow movement of material starting from the mouth, so we need to have swallowing, and then taking the long time to go through the various organs of digestion, the stomach, small and large intestines. And this is helped by the movement of the contraction of smooth muscle to move things from one part to the other. In order for digestion to occur, enzymes are required, and enzymes are produced and released in a few parts of the digestive system, allowing it to break down various products. We'll talk about a couple of enzymes, a couple of hormones, that help regulate digestion in the absence of the nervous system. And then finally talking about absorption, primarily in the both small and large intestines. Now the digestive tract usually has some sort of immune barrier so that all that external microorganisms that you take in, yes, you take in lots and lots of bacteria with every biteful, does not have to react too much with it. Now there are some immune system cells that can get into the gut, but primarily the innate immunity that keeps these possible pathogens from entering the rest of the body is done via enzymes like lysozyme, and of course the pH changes that we'll see, especially in the stomach. The digestive system is divided into a wide range of organisms, and we'll talk about all of these listed in this slide as we move throughout the lecture. And here is the placement of all these various organs, starting with the mouth and ending, of course, with the anus. Are you ready for our journey down the alimentary canal? All right. Feeding takes place in the general organ known as the mouth. The mouth is there to bring the food in, but the mouth is also appropriate for beginning the digestive process. And so as you bring it in, your tongue helps manipulate the food around the mouth. The food is lubricated with the help of saliva, which not only contains lysozymes, which we discussed for exam 3, but also the first enzyme of digestion known as amylase. Amylase is an enzyme that begins the process of breaking down starch into the disaccharide known as maltose. Finally, the mouth contains lots and lots of teeth, and the teeth is there to break up the large pieces of food into smaller pieces of food to increase the surface area so that the subsequent enzymes, including salivary amylase, can begin to break down the larger products. After food has been lubricated, after food has been chewed into nice small little bits, you're ready for one of the most interesting muscle contraction, muscle coordination events in your entire body: swallowing. Swallowing has basically a couple of parts. The first part is voluntary. You voluntarily chew your food. You voluntarily, with your tongue, move the food towards the back of your throat. The second part is involuntary. You have no control over this except to start it. So you can hold food in your mouth forever and you will not start the process of swallowing until the food is placed to the back of your mouth. Then once it starts, all of these muscles must work in kind of a coordinated, synchronized manner to do various things to allow the food to move from mouth into the second part of the digestive system, the esophagus. The esophagus, once it receives the bolus of food, uses smooth muscle to contract in a peristaltic manner to move food down into the third organ of digestion, the stomach. And I just swallowed a nice big gulp of water. It's just seeing the movement, the contraction of smooth muscle to move the food, the bolus, into the stomach. Okay, the stomach. The stomach has primarily two functions. One we discussed in exam 3, and that is an immune system function to kill most bacteria that enters your body via the mouth. It does this because of a very, very high acidic secretion of hydrochloric acid. But this secretion of hydrochloric acid also enables the stomach to conduct its second function, and that is protein digestion. The food will remain in the stomach for, depending upon the size of the meal, up to 2 hours. And during that 2-hour process, there will be gastric secretions, material moved out of the stomach, which we're going to talk about in a few slides. Also makes very good exam 4 questions, by the way. And allows the process of mixing the bolus with the one enzyme found in the stomach, pepsin. Pepsin is a protease and breaks down proteins into smaller pieces, even amino acids. This slide shows you the cross-section. The internal structure of the stomach is just lined with the kind of endothelial tissue. But there are cells that will secrete various substances that are quite important in the working of the stomach. Okay, so let's go down the list. There are chief cells. The chief cells secrete a proenzyme called pepsinogen into the contents of the stomach. Pepsinogen will convert to the active form pepsin, that protease that breaks down proteins in your stomach, because of the actions of hydrochloric acid. Hydrochloric acid, secreted by parietal cells, is secreted for 2 reasons. One, well, actually 3. Let's do it 3 reasons. One, of course, immune system function, kill bacteria. Bacteria do not like a pH of between 1 and 2. It's not good for them. It's not good for anything. Primarily, it gets into your stomach. Yes, there are a few things that can get through it. Second function of HCl is to create an acidic environment so that the proteins that are placed within it hydrolyze a bit. They unwrap, they unfurl to allow the protease, pepsin, to break them down. Another type of cell found within the stomach are goblet cells. And goblet cells secrete mucus. And the mucus is there to protect the inner lining of the stomach from that high acidic conditions. So, chief cells secrete pepsinogen, parietal cells secrete HCl, goblet cells secrete mucus. How is that possible? It's a timing mechanism initially controlled by your brain. Okay? You are sitting in your car, in your bedroom, wherever, outside, sunning yourself, listening to this slide. I want you to think about your favorite breakfast, lunch, dinner, depending upon the time of the day. Got it? Think about it. Tastes good, doesn't it? Just by thinking about it, or seeing it, smelling it, tasting it, hearing the pan of bacon sizzle is enough to stimulate the vagus nerve, that 10th cranial nerve, remember that one? To stimulate the chief cells to secrete pepsinogen, parietal cells to secrete HCl, and the goblet cells to secrete mucus in preparation for lunch coming down the pike. Okay? So, the initial activation of the stomach to begin the process of digestion is the nervous system. But as we know from multiple lectures, especially during that exam 2 phase, that the nervous system is short-lived. It doesn't last very long. But yet the stomach, depending upon the size of breakfast, lunch, or dinner, or midnight snack, will spend up to 2 hours dealing with your midnight snack. The nervous system doesn't last that long. So, after a few minutes, the nervous system's activation is replaced by the endocrine system, specifically the hormone, peptide, called gastrin. Gastrin is secreted by what are called G cells, also found within the stomach. This cross-section, there are these deep gastric pits. This is where the cells are found. The goblet cell, the parietal cell, the chief cell, all secreting the various ingredients that make up stomach digestion. Again, to reiterate, the functions of HCl, again, to make the stomach contact more acidic. This acidic content denatures proteins to allow the pepsin, the activated form, to function and to break down bonds much easier. HCl also helps convert the inactive form pepsinogen to the active form pepsin. If you remember way back from lecture 4, we're talking about enzymes and their optimal pH. Well, the optimal pH for pepsin is between 1 and 2. And of course, HCl is part of the innate immune system. Okay, we've talked about this one, that the conversion of pepsinogen to pepsin does not require a second enzyme. Instead, it is simply the presence of hydrochloric acid, HCl. Let's go back to gastrin. Remember, gastrin is a peptide hormone that is released by the G cells, which are also found in the gastric pits, much like right next to the parietal cells and chief cells and goblet cells. Gastrin's release is stimulated by the stretch of the stomach. So again, kind of a proprioception activation. So the presence of food within the stomach provides weight or stretch to the stomach, and that is the stimulus that causes the G cells to release gastrin. After the vagus nerve stops activating the cells, gastrin takes over, as these cells have receptors for gastrin on them, the parietal cells and the chief cells and the goblet cells. so that gastrin can continue activation of the stomach digestion as long as the stomach is stretched, as long as the food, the bolus of food, remains in the stomach. This is considered a positive feedback, and we'll talk about positive feedback and negative feedback in the next lecture of basic endocrinology. All right, so we've talked about chewing, swallowing, and stomach, literally protein digestion. It's time to talk about the real workhorse of the digestive system, the small intestine. Small intestine can be as long as 30 feet in the human. Anatomists can have divided the small intestine into 3 sections. The only one we're really going to talk about is the first one, the duodenum, because that's where all the small intestine digestion, at least initially, begins to take place. Both the jejunum and the ileum are there mostly for absorption. Smooth intestinal cells are replaced very often. It's one of the more highly mitotically active cells, constantly being produced and removed. Small intestine will have increased surface area because of both the villi and within the cells, the microvilli. All of this increases the surface area that increases absorption of the smaller bits, the end result of digestion. Whereas the stomach digestive functions, at least, were just the breakdown of proteins, the small intestine is all of the above. All digestive enzymes for everything, carbohydrates, fats, proteins, other, are found within the small intestine and are released within the duodenum. The absorption of all the breakdown products takes place in the ileum and the jejunum. So the gross structure or internal structure of the smooth intestine has lots and lots of these villi, these finger-like projections in which the cells are part of. Again, function of the villi is increase cell surface area, increases absorption. Now within the cells, each individual cell may have its own villi, known as the microvilli. We talked about that probably in lecture number 2. The microvilli have the ability to kind of wave, to help kind of produce some sort of microflow, microenvironment. And that waving is caused by our 2 best buddies, actin and myosin. But also deep within the villi itself are these smaller little caves or crypts. Your book calls them intestinal crypts. My favorite word is they're also known as the crypts of Lieberkühn. Sorry, didn't know where to put the umlaut over the U there. And this is where the duodenum itself will produce digestive enzymes and lysozyme to attack that bacteria that snuck through the stomach. Again, this is just a cross-sectional cartoon looking at the cross-section of the villi. You see the villi are made up of multiple cells. Each cell is very close to a capillary for absorption. Within the duodenum only, there are the crypts of Lieberkühn, labeled here intestinal crypts. And these are the cells that will secrete duodenal enzymes. Here's a few of the small intestine enzymes produced by the crypts of Lieberkühn, the intestinal crypts. You see all sorts of it over there. The intestine also produces, again, analogous to the stomach, a positive feedback hormone known as cholecystokinin. That is one huge word. Forget it. Let's just call it by its nickname, CCK. Also released by the intestinal crypts, CCK also takes over the actions from the vagus nerve to continue the digestive process within the small intestine. So, 1, it continues the activation of enzymes from the intestinal crypts. CCK will also activate the gallbladder to release bile. We'll talk about the gallbladder in just a little bit. CCK will also activate the pancreas. Pancreas is an digestive organ that will increase the amount of enzymes, pancreatic enzymes, necessary for the completion of digestion within the small intestine. CCK will also, it's going to get long, stop stomach contractions, stop stomach digestion, primarily by blocking the release of gastrin. Finally, CCK, interestingly, has receptors in the brain, specifically in the hypothalamus. CCK's function? Satiation. I'm full. Where does that happen? Oh yeah, let's wait to the end of our today's lecture, as when we talk about control of feeding. Okay, so cholecystokinin, CCK, secreted by the duodenum as soon as the bolus of food enters the duodenum. CCK is a smaller peptide, and there, as you can see, the functions active as secretes enzymes, both from the intestine and the pancreas. Activates the gallbladder to release bile. Blocks gastrin production within the stomach. And appears to play a role in the cessation of feeding by activating parts of your brain, the hypothalamus. So let's talk about 2 other organs of digestion, the liver and the pancreas. The liver has a lot of function that we've been talking about last couple of weeks especially. It produces a lot of stuff for the blood. Complement. A lot of the clotting factors. The blood protein albumin, all produced by the liver. But the liver also has a digestive system function, and we'll talk about that in just a little bit. Just simply a chart of what the heck the liver can do for you. So there's all sorts of things. We're going to concentrate on the last category, and what the hell is bile? There is an interesting connection between the liver and the small intestine, and it is known as a portal system. Portal system, the anatomists will describe it as a capillary system between two veins. Okay, so the hepatic portal vein is a vein that goes from the small intestine to the liver. So it can bring nutrients into the liver for processing, storage, perhaps even detoxification. Okay, so the liver can connect with the small intestine in two ways. First of all, outflow, nutrients moving from a capillary into the hepatic portal vein into another capillary bed so that the liver cells can process these nutrients. But the liver is also going to provide extracts, aids for digestion via the gallbladder. The gallbladder is actually part of the liver. It's a little storage sac surrounded by smooth muscle, which when contracted secretes its product known as bile through the common bile duct into the duodenum. Okay, so a little brief words on the gallbladder. So it's going to store bile, which are secretions coming out of the liver. We'll talk about bile over the next couple of slides. Eventually, the gallbladder via the common bile duct will release its products, bile, into the duodenum for processing or for use in digestion. Here are many of the compounds found within the bile duct. You'll see at the bottom, you'll see breakdown products because of drugs that you've taken. Again, remember the function of the liver, detoxification, and it excretes a lot of the material that it secretes via the gallbladder. Well, we are going to talk about two products produced by the gallbladder, or released by the gallbladder, bile salts and bilirubin. So there is approximately half to 1 liter of bile produced every day, all entering the small intestine at the duodenum. So we're going to talk about two products released by the gallbladder into the small intestine. The first one is bilirubin. Bilirubin is the breakdown product of hemoglobin, specifically the heme group. Remember the heme group, that kind of organic molecule in the middle of hemoglobin with the iron molecule in it, and which is where the oxygen attracts. Remember that? Exam 3? Anyone? Okay, you don't need to know it for exam 4. But bilirubin is a breakdown product of heme. This breakdown occurs within the liver. Bilirubin can be slightly toxic and is stored within the gallbladder for storage until it's put into the intestinal tract. Interestingly, if you have too much bilirubin around, your skin will turn yellow. There is a affliction known as jaundice, which can occur because of a wide range of different ailments, from liver disease to blockage of the gallbladder, stones. Another interesting fact about bilirubin is when you get bruised for the first time and your skin turns that disgusting little yellowish phase, that is from bilirubin and the disruption of red blood cells within the bruising area. Bilirubin, 2 more points. One, we'll talk about bilirubin during childbirth. Remember in exam 3, we talked about that fetuses have a different type of hemoglobin than post-birth adults, I guess air-breathing things, right? And within the first couple of months of the newborn's life, it needs to break down the fetal hemoglobin so it can produce adult hemoglobin. Breaking down hemoglobin relies on the liver, which there's going to be extraction of the bilirubin, breakdown of heme to bilirubin, and then extraction of it. And the first thing the neonatal doctor will do is test to see the bilirubin concentration in the newborn. If it's too high, usually they don't allow the child to go home until the bilirubin levels decrease. And this can happen 2 ways: one, by drugs, or even better, you put the young kid, Southern California, of course, has to go into the tanning salon. It's Southern California, right? Well, kind of. UV light is another way to break down bilirubin, to make it less toxic, because bilirubin in high levels has been shown to produce neuronal damage in the neonate. So why do they test bilirubin in the adult, in the newborn child? Is the liver functioning, yes or no? Final part about bilirubin: yes, it is bilirubin that causes your feces to have that nice color. The last chemical to talk about within the gallbladder and the secretion of bile into the small intestine is a digestive aid known as bile salts. As the name implies, we are going to add salt, and we are going to add salt to cholesterol. So salty cholesterol. Function? Okay. This one you only have to memorize one word, and that word is emulsify. The function of bile salts is to emulsify fats. What the heck does that mean? All it means is make the fats more water soluble. So, water soluble enzymes like lipases can break them up. How does adding salt, sodium, to cholesterol make it more water soluble? This question may sound kind of off the wall, but what the heck? How do you make soap? No, you don't, not going to Costco and buying in bulk. No. I've been making it, not buying it. You take animal fat and you add salt to it. So basically, bile salts are analogous to soap to help emulsify. I don't want to say break down, make fats more water soluble. I don't know why they're showing you the thing, because that's cholesterol. There's no salts attached to it. All right, our second organ accessory. You can see this. So the pancreas sits sandwiched between the stomach and the small intestine. Again, it has two functions. One is hormonal, but the other one is digestive, specifically digestive enzymes. This table shows you many of the digestive enzymes produced by the pancreas. The most famous one, of course, is trypsin. Trypsin is produced by the pancreas and released into the duodenum. Another molecule that is produced in the pancreas and released is bicarbonate. It's not an enzyme. Why would the pancreas release lots and lots of bicarbonate into the duodenum? Remember the function, one of the functions of bicarbonate? A buffering agent for the blood, or in this case, a buffering agent for the duodenum. Reason? Remember all that stuff coming out of the stomach? It's very acidic, isn't it? Bicarbonate helps neutralize the HCl produced by the stomach. Oh, sorry, just taking a big glass of water, which is important for this last section of the digestion tract, the large intestine. Here are some of the parts of the large intestine. Here is a picture showing where the parts are found. Large intestine is about 6 feet in length. It is considered large because of the diameter, not because of the length. And the large intestine has a few but interesting functions. Number one, the majority of salts, sugars, amino acids, fats, etc., are absorbed within the small intestine. But yet there are things that are absorbed by the large intestine, primarily water. Works via osmosis to move into the blood. But there's also absorption of potassium, ions, and also some important vitamins. Important vitamins produced by our symbiotic microorganisms, bacteria. Yes, you have lots and lots of different bacterial species growing within your large intestine. Don't be alarmed. The majority of them are harmless and incredibly important. So the microflora, there are hundreds of different species that have been found within humans. You may have up to 100 different species yourself. Most bacteria in your guts are not harmful. There are some that are, E. coli being one of them. But E. coli within your large intestine, they're fine. It's when it gets out of the large intestine that there can become a problem. The bacteria living within your large intestine survive like any other microhabitat. They'll survive. Most of them are anaerobic. They don't do well in the presence of oxygen. And you always have it. You have it because of all the food you ate and you will eat. Nursing from mom, right? Because our bodies are filled with bacteria. Chewing on toys, whatever. So why do we have bacteria? Well, number one, to produce methane gas. Sorry, that's not a benefit. But many bacteria within our digestive systems will produce essential B vitamins that we cannot produce by ourselves, nor can we get from our foods. So we have this nice mutualistic arrangement. We provide the bacteria with room and board and a constant temperature. What do they give us? Essential B vitamins. Now, as you live your daily lives and you decide to eat the pizza last night or the avocado and cheese sandwich or the whatever, your microflora will change over time. So you start out with one. By the end of the week, depending upon what you are eating, or in some cases drinking, as consumption of alcohol can alter the population. So these populations are going to change. Some will benefit, some will not. Which leads us to probiotics. You've seen a lot of ads on TV, going to health stores, the benefits of probiotics. Probiotics will alter your entire body by absorbing different bacteria. It is true that as your bacterial flora, microflora will change, you can feel the digestive effects of it. Some good, some not so good. So is there a benefit with probiotics? Trying to alter your microflora. I'll let you figure that one out. All right. So we've talked about digestion. Actually, there isn't much physiology in digestion if you think about it, right? It's mostly biochemistry and anatomy, and with a little hint of microbiology. So the fun part is, let's talk about the regulation of feeding. I bring this up since no other human physiologist will bring it up, simply because this is part of my PhD thesis. I was interested in the regulation of feeding by caterpillars. We won't go into that. Instead, we'll talk about the regulation of feeding in humans. Ready? Being the neurobiologist that I am, we are going to have to talk about the brain and how the brain controls feeding. Primarily, let's isolate our lecture on one part that is known as the hypothalamus. You can see that on the left hand of the screen, pointing to a structure kind of at the bottom of the brain. Right? We've already talked about the hypothalamus in terms of release of antidiuretic hormone. The hypothalamus we already talked about. Remember fever and endogenous pyrogens? Remember that one? So the hypothalamus controls all your drives. So hunger, thirst, temperature, sex, etc. And it plays a huge role in how we feed. It's a cross section. The cartoon on the right kind of, again, shows you the temperature control and thirst and hunger and all that fun stuff is controlled. We'll talk about the hormonal structure that it activates the pituitary really soon in the next couple of, actually the last three lectures of the course is you and your pituitary, you and your hypothalamus. But let's talk about you and your hypothalamus when it comes to feeding. Please take a note of 3 different regions of the hypothalamus. First one is the ventromedial hypothalamus, or the VMH. You can see it there. Right next to it, in the kind of the, whatever color is that, bluish green small little oval, is called the paraventricular nucleus, or the PVN. And then on the outside of the hypothalamus is a place known as the lateral hypothalamus. See 2 regions over there? That's right. All 3 regions play a role in hunger. Hunger, by definition, is the obsession to eat. I must eat. I'm hungry. There's our 3 regions in the screen here. The lateral hypothalamus, or the LH, the PVN, not pictured here, but still. Those 2 are known as the hunger centers. The VMH, or the ventromedial hypothalamus, is the satiety center, or I'm full, I don't want to eat anymore. Some 50 years ago, more than that, 50 to 60 years ago, evil scientist would take a poor helpless rat and ablate these 3 areas. Ablate means basically lesion it, cut it out. Okay? There's an alternative you can do, is instead of sticking something in there to chemically ablate it, or now they use lasers to do these things, etc., you can stick an electrode and you can stimulate these 3 areas as well. Okay? So let's talk about the hunger centers, the LH and the PVN. Lateral hypothalamus, paraventricular nucleus, the hunger centers. Okay? If you stimulate these 2 regions, either separately or together, doesn't matter, you stimulate it, the animal overeats and becomes fat. If you remove by chemical ablation or lasers, surgery, whatever, the LH and the PVN, the animal does not start to feed. Got it? Hunger centers. The VMH, the ventromedial hypothalamus, is the opposite. If you stimulate the ventral medial hypothalamus, the animal, you can see in the upper part of the screen, if you stimulate the VMH, you do not get the animal to feed. The animal won't start feeding, right? It stops feeding. I'm full. It always has that sensation that I'm full, I'm full, I'm full, I can't eat. Okay? If you were to lesion the VMH, remove it. The animal cannot stop feeding. It overeats. Got it? So, you have this interesting dynamic between the hunger centers and the satiety centers. When to activate each is, okay, I'm hungry. Does that mean the LH or the PVN get stimulated? Or does that mean the VMH get inhibited? Good question. There are probably at least 1,000 neuro-nerds in the world studying just that question. Okay? But what about appetite? Appetite is defined as a specific hunger for a specific nutrient. Right? I want to eat sugar. I don't want to eat sugar. I want to eat something fattening. I don't want to eat something fattening. Correct? Do these three regions, the VMH, the LH, and the PVN, allow you to select the proper meal during pick a time, breakfast, lunch, or dinner? These studies are known as cafeteria studies, diet selection, nutrient self-selection, and have their basis because of a study done with pigs at Iowa State College of Agriculture, which was shortened about 100 years ago to Iowa State University. There, one of the techs, a guy by the name of James Everett, he finally got his PhD at the University of Missouri, I think, and then came back and continued teaching at Iowa State. Way back when, we're talking about the early 1900s. He was a tech. Okay, he was a pig tech. He raised the pigs for the university to play with, have their bacon fried before Iowa State football games. Who knows what he did then? But he was very interested in how his pigs ate. Sure, he'd give them the Purina pig chow, but he noticed that his pigs would eat other things. And so in around 1910, he decided to produce what is known as a cafeteria study, where he allowed his pigs to grow on a choice of food. He obtained a nice large litter. I believe it was a litter size of, 8 or 10, half of which the little piglets, after they were weaned from mom, ate only Purina pig chow, which of course stupid humans thought pigs liked the best. And the other was given a bunch of choices of oats, corn, wheat, whatever pigs eat. But he gave him a choice. And all he did, he didn't measure what they ate. All he measured was their weight gain. And unbelievably, or maybe believably, the pigs that were allowed to choose their own complete meals grew significantly better than the pigs given the smart human pig chow. Wow, pigs are smarter than humans. No comment. One interesting sidelight from Everard's study in 1910 was up until 1980, and I've lost track, one of his sows for us Southern California urbanites, that is a female pig. One of the sows still was the largest. One of the sows in his study was still the largest sow ever grown in the state of Iowa. Exciting. So you can see he published his studies, and it took a while because it was in this obscure agricultural journal of Iowa. But it got the word out that this pig was able to do a self-selection, a diet choice, a cafeteria experiment. Can other mammals do it? And in the late 1910s, up into the 1920s, and still today, that's why I got my PhD, showing that caterpillars can do it. But you got all these scientists working on diet choice. So you see it in mice and rats and dogs and cats and sheep and goats and cattle and chickens and lizards, and all showing their ability to self-select a proper and well nutritious diet for themselves. So pigs were not the exclusive one. All mammals seem to do it. Well, what about humans? Can humans self-select their own diet? Well, it didn't take long for psychology, nutrition departments to begin these studies. And after, decade and a half of experiments, it was shown that humans, no, they don't self-select a good diet. So, we're superior. Wait a second, we're mammals. We basically eat the same things that mammals, like pigs, eat. So why couldn't these studies prove this? Well, university studies, I wonder where they get their subjects for these studies. You are correct. College students. Shall I even question the ability of a college student to eat a good, nutritious diet? See, the problem with college students, adults, even children, because of cultural things and chemical things and physiological states, by the time a college student hits campus, they already have a preconceived notion of what's good and what isn't good. And so there's a lot of bias that goes through human food choice. But what if we can get some sort of naive human? Yes, that's me, feeding my daughter at age 3 months her first non-breast milk formula food, cereal. She seems happy. In the late 1920s, a pediatrician, who was interested in nutrition, by the way, in Chicago, got very interested in this food choice experiment. And she theorized that the reason why humans were not shown to have the ability to make these dietary choices was because they were they already had a preconceived notion of what foods are and what's food good for them based upon all sorts of things. But what happens if you try this with young children? There she is, Clara Davis. She was a pediatrician in Chicago in the late 1920s. And you can see a couple of papers that she wrote, one early on, and then a much more broader one. The first one done in 1928 was done actually with six small little boys, which we're going to describe this. Now, remember what's happening in the late 1920s? Oh yeah, the Great Recession. Excuse me, not recession, depression. Okay, so because of that, there were a bunch of orphaned kids about, and she actually selected, because they were ill, 6 of them. These were between 4 and 6 months old, and all they had was bad gruel to eat. What happens if she gave these children the choice? This is from that first paper. This is a photo showing the choices that the children got. Looks yummy, doesn't it? Okay. Here's the procedure. She instructed all the nurses to bring a tray of food, much like this one that you see, to the volunteers. Right. These 6 little boys, again, ages between 4 and 6 months old. The nurse would present the tray to these research subjects. These little boys would then pick what they wanted to eat. The nurse was instructed to take a spoon and feed them what they wanted until they didn't want it anymore, pointed at something else, and feed them. And all the nurse did was keep track by weight of what foods they ate over the course of a couple of months. I think, I don't know, it was 3 to 6 months on most of them. Okay. Here are the results. Here are 3. This is Earl, Abraham, and Donald. You can see the kind of the pie diagrams of the percentage of food each kid ate. Remember, this is the late 1920s. Dr. Davis did not have Excel at hand. I guess the hospital didn't buy it for her in 1928, and actually drew these graphs freehand. Okay, so if you take a look, you'll see that these kids ate at least kind of the proportions of especially the fruits and cereals and milks, at approximately the same rate. She also measured the weight of these children, and you can see from one ingredient, one nutrient that seems to be established to help you control is carbohydrate, and that is through the neurotransmitter serotonin. When you are taking lots and lots and lots of sugary-based foods, like the orange juice and the waffle, you increase the levels of brain tryptophan. Tryptophan, if you recall, is the precursor to serotonin. Increases serotonin levels, causing you to reduce carbohydrate hunger, appetite. The opposite is true. If you take things that are low in sugar, your tryptophan levels will drop, causing you to perhaps want to have more carbohydrates. Here's a study on humans, given by the serotonin carbohydrate team of Wortman and Wortman. Mixed marriage. Judy was an MD. Richard was a PhD. It was Wortman's lab that initially discovered the link between serotonin and carbohydrate way back in the 1970s. This study is showing a experiment involving humans and taking a drug or a placebo. The drug is a mix of two drugs called fenfluramine and phentermine, or fen-phen, which used to be the best dietary drug in the US until they discovered it had lots of lethal side effects, and it's been banned from the US. Fenfluramine works like Prozac. It's a serotonin reuptake blocker, which increases the level of serotonin in your brain. Remember, high levels of serotonin make you eat less carbohydrates. Given the fact that 80% of our diets are carbohydrates, if you don't want to eat a carbohydrate, you eat less, and you can see a significant drop in weight among humans. Here's the last study, again by our best buddy Dick Wortman. This is injecting serotonin, 5-hydroxytryptophan, into rats and looking at the food intake. The more serotonin, actually there's a serotonin precursor into it, the less the rats eat. Does this go on in humans? Are your serotonin levels determining what you eat? Happy feasting!